Hi everyone, this video is an introduction to a very important macromolecule, that is nucleic acids. The monomer of nucleic acids is the nucleotide. There are three components that I'd like you to know about the nucleotide: the nitrogenous base, the pentose sugar, and a phosphate. The sugar and the phosphate make up the backbone, and the nitrogenous base makes up the genetic coding part of the molecule. Carbon residues, in the pentose sugar are labeled 1 through 5 prime. Here would be 1 prime, 2 prime, 3 prime, 4 prime, and 5. The base is attached to the sugar at position 1, shown here. The phosphate is attached to the sugar at the 5 prime position, shown here. When a polynucleotide forms, the incoming nucleotide's 5 prime phosphate attaches to the 3 prime hydroxyl group at the end of the growing chain here. So you'd see another incoming nucleotide joined together here to this nucleotide. The naming convention of 5 prime to 3 prime is also how we get the orientation of the molecule, which I'll get back to in a moment. There are two types of sugars in nucleic acids. In DNA, it's deoxyribose sugar, and in RNA, it's ribose sugar. The only difference between these molecules is that deoxyribose has a hydrogen here at position 2, whereas ribose has a hydroxyl group here at position 2. We can divide bases into two categories: purines and pyrimidines. Purines have a double ring structure like those shown here, whereas pyrimidines have a single ring structure like those shown here. The mnemonic device that I use is that cytosine and thymine, both of which contain a Y in the spelling, are pyrimidines, and pyrimidines, of course, also has a Y in the spelling. Uracil is also a pyrimidine, which is substituted for thymine in RNA molecules. So DNA is a double helix. So it's composed of antiparallel strands. So it's made up of two strands that are twisted into a helix. The phosphate backbone, shown here in this image as the curvy lines, is on the outside of the molecule, and the inside are the bases, base pairing. If we were to straighten out this molecule, it would essentially look like a ladder, wherein the sugar phosphate backbone would be the handrail here and here, and the nitrogenous bases base pairing would be here in the middle, forming the rungs. Each base from one strand interacts via hydrogen bonding with a base pair from the opposite strand. That's easily seen here in this image. So as I mentioned before, DNA is antiparallel, which means it's running two strands in opposite directions. So you can see from this image here, this would be the 5 prime end of the molecule for this strand, and this would be the 3 prime end. If we look at the same orientation, the opposite strand would be running 3 prime to 5 prime in that direction. Let's look at this at a more simplified level. So here you have the 5 prime end of the molecule, and here's the 3 prime end, whereas the opposite side, the other strand, is running in the opposite direction, 5 prime to 3 prime in this direction. Okay, so that's what I mean by antiparallel. We will discuss DNA's antiparallel nature in greater detail when we cover DNA replication and gene expression down the road, so don't worry about that too much at this point. Okay, so like I said, the phosphate backbone is on the outside. That would be this shown here and here, whereas the bases in the middle form the rungs. Okay, so adenine forms hydrogen bonds with thymine. Guanine forms hydrogen bonds with cytosine, and that is the convention for base pairing when it comes to DNA. Now, the base pairs that are occurring between these nitrogenous bases on the two strands of DNA are weak hydrogen bonds. So here's 2 weak hydrogen bonds between thymine and adenine, and here's 3 hydrogen bonds between guanine and cytosine. So they're weak to the point where if you heat this molecule up, you can actually separate the two strands that make up the double-stranded DNA molecule. So again, just as a reminder, you get 2 base pairs between T and A, and you get 3 base pairs between C and G. So what does DNA do? What does RNA do? Well, they both participate in a process called gene expression. So in this case, DNA acts as a genetic code for constructing RNA. RNA acts as a code for constructing proteins. Now, we're not going to go into the details of gene expression just yet. This picture is showing the process of translation. We're not ready to discuss that yet, but you really should understand that DNA, deoxyribonucleic acid, is the genetic code which makes all of the proteins ultimately that we've discussed. So it's the sequence of these nucleotides that determines ultimately the structure and function of the proteins that we've discussed in class. Okay, so I hope this helps as an introduction.